Hi kids, so it's been a while since I've made a video. In this video particularly, we are going to continue in the series of general pathology videos, and today I'm going to cover an important topic from the chapter of Neoplasia, which is tumor suppressor genes. What are tumor suppressor genes? The products of most tumor suppressor genes, as the name indicates, they are genes which are suppressing the occurrence of cancer in your body. So they're suppressing the occurrence of tumor in your body. Whenever a tumor suppressor gene is activated, it will apply brakes to the cell proliferation, and if there is an abnormality in the tumor suppressor gene, it will lead to a failure of growth inhibition. That is, growth inhibition that is normally being controlled. The growth is normally being controlled. Normally our cell cycle is tightly regulated, and that fails, which is a hallmark of carcinogenesis. Tumor suppressor genes control a series of checkpoints in the cell cycle, which will prevent uncontrolled growth. And the two main genes that are involved in this are RB and P53. Now we'll discuss these two. And RB and P53 are two important topics for short notes, both in UG as well as PG exams. Let's go. So, of course, there are many genes involved apart from retinoblastoma gene and P53, but these are the most important ones, so we're discussing them. RB gene is a key negative regulator of the G1S cycle transition and is directly or indirectly inactivated in most cancers. It's the first tumor suppressor gene to be discovered. It was identified by studying a rare genetic disorder called retinoblastoma, and hence its name, which is a childhood tumor. Approximately 60% of the retinoblastomas are sporadic, while the ones which are familiar are inherited with an autosomal dominant trait. So there's a scientist called as Knudsen. There was a scientist named Knudsen. He gave a two-hit hypothesis for the appearance of retinoblastoma. He said both of the genes of retinoblastoma have to be mutated in order for retinoblastoma to occur, whether it is the sporadic occurrence or it is a genetically acquired autosomal dominant inheritance. These mutations, these hits are for RB gene, which is located in chromosome 30, Q14. Both of the normal alleles of the RB locus must be inactivated, that is, two hits, for the development of retinoblastoma. So, if it's a genetic case, the autosomal dominant inheritance one, the child is going to inherit one defective copy from one parent, but the other copy is still normal. So the RB is still normal, but retinoblastoma will develop when that normal gene is also mutated, somatically only. In sporadic cases, both of the genes are lost because of the somatic mutation. So the end results of both of these is the same, that a cell that has lost both of the normal copies of the RB gene will become cancerous. Now we'll know why. Now we'll get to know why does that happen. This is just a symbolic representation from Robbins of the pathogenesis of retinoblastoma, whether it is sporadically inherited or if it is genetically inherited. Now, you know what is cell cycle, right? We've read this since plus 1, plus 2. Cell cycle involves four phases, G1S, G2M. They are important checkpoints. So G1 is growth and S is DNA synthesis. Then is G2 phase, where there is preparation for mitosis. And then M is where finally your cell is going to divide. There's some things which make sure that this cycle keeps going. These are written as GO. These are growth factors, oncogenes, and cyclins and CDKs, cyclin-dependent kinases. So growth factors, your oncogenes, and cyclins and cyclin-dependent kinases make sure that your cell cycle keeps going. The things that stop it, because we have a tightly regulated cell cycle, right? So the things that stop it are tumor suppressor genes and CDK inhibitors, that is cyclin-dependent kinase, are inhibited by some things called as CDK inhibitors. So the tumor suppressor genes and your CDK inhibitors are important for you to not have cancer. Now, we know that there's a G1S transition where there's a checkpoint, G2M transition, there's a checkpoint. Once the cell has entered the S phase, that's the point of no return. Delay in cell cycle progression provides the time needed for DNA repair. So whenever there's a checkpoint, there's a hold. That's because there's some damage has occurred in the cell, and in that we can that body makes sure that the DNA repair is occurring. We'll see how that also happens. If the damage is not repairable, So the cell will have to be killed by apoptosis. G2M checkpoint monitors the completion of the DNA replication which is happening in S phase, particularly important in cells which are exposed to ionizing radiation. Defects in these checkpoint components is a major cause of genetic instability in cancerous cells. Now, let's understand how RB is playing its role in the G1S checkpoint. How is it acting? And if RB is not there, how is the cell dividing? So, there is something called, let's start with growth inhibitors. Okay, whenever your cell is dividing, your cell was dividing normally, and now we want to stop that. We want that the cell should not divide anymore. We are done with its function. So, growth inhibitors, that is TGF beta, P53, etc., they will come and they will promote the formation of CDK inhibitors like P16. These CDK inhibitors, we have read about CDK inhibitors if you've seen the chapter of cell injury. CDK inhibitors will inactivate complexes. These complexes are formed of cyclin D, cyclins and cyclin-dependent kinase. Remember in the last slide I told you that the cyclins and cyclin-dependent kinases make sure your cell cycle is going. So these cyclins and cyclin-dependent kinases, they form certain complexes. And your CDK inhibitors are stopping these complexes. The stopping of these complexes will lead to the hypophosphorylation of RB. RB has two forms, activated form and inactivated form. If you look at this picture, hypophosphorylated form has only 2 phosphates, but the hyperphosphorylated form has 6 phosphates. That's the difference between the phosphorylation. But the active form of RB is the hypophosphorylated form and not the hyper one. This active RB will attach to the E to F transcription factor. Now, if you remember the pathway, how does a replication happens? You have transcription, translation, and all that, right? If you remember your +1, +2. So, उसमें elongation factors होते हैं. There's a promoter site, there's an elongation site on the DNA, and E to F site को E to F ने आके लगना है. E to F site पे E to F elongation factors ने आके लगना है. But active RB ने E to F को अपने पास पकड़ के रख लिया है. तो आपकी ट्रांसक्रिप्शन हो ही नहीं रही. Okay? So let's read. The initiation of DNA replication requires replication होने के लिए it is needed that your cyclin and cyclin dependent kinases का जो complex है वो active रहे, because if they are active then only your E2F family of transcription can continue with the transcription. क्योंकि replication से बाद transcription उस रेप्लिकेशन होगी जीन की, बट उसके बाद उसका ट्रांसक्रिप्शन यानी कि जीन का प्रोडक्ट जो mRNA बनता है वो दैट इज इंपॉर्टेंट, राइट? ट्रांसलेशन और ट्रांसक्रिप्शन आर वेरी इंपॉर्टेंट प्रोसेसेस. दे हैव टू फॉलो द डीएनए रेप्लिकेशन, अदरवाइज सेल आपका डिवाइड नहीं कर पाएगा. So that transcription is stopped by RB. Early in the G1 phase, अब checkpoint पे बात कर रहे हैं, G1S checkpoint पे, early in RB in its hypophosphorylated active form will bind to and inhibit the E2F family of transcriptors, and then thus it prevents the transcription of cyclin E. This is normally how RB is saying कि इस checkpoint पे I think we should stop division of the cell, G1S checkpoint पे. But in the opposite case, if the cell had to be divided, so it is normally जब भी cell division हो रही है किसी cell में, या फिर even cancerous cell में भी यही mechanism है कि growth factors आए, like epidermal growth factor, platelet growth factor, उन लोगों ने आपके cyclin dependent kinases और जो cyclins का जो complex है उसको activate किया. Remember they are needed for the cycle to continue, and उन्होंने आपकी जो RB है उसे inactivate कर दिया, hyperphosphorylate कर दिया उन kinases ने. अब E2F is free. E2F is free to bind to its E2F site and transcription continues and the cell will enter the next phase. Hypophosphorylated RB blocks E2F mediated transcription in these two ways. Firstly, it will sequester E2F to itself and it will prevent it from interacting with other transcriptional activators. Secondly, it also recruits chromatin or remodeling proteins like histone deacetylases and histone methyltransferases, which will bind to the promoters of E2F responsive genes such as cyclin E. So it is not just controlling E2F directly, but it is also controlling the chromatin remodeling proteins. So indirectly, it is also controlling E2F. This is the same cycle. Now, we know that oncogenic viruses are also causing their cancer by inhibiting RB. We just know कि अगर damage है किसी DNA में तो RB will stop the G1S checkpoint in order to make sure कि वो division रुक जाए. लेकिन जब oncogenic viruses आ रहे हैं, they are stopping They're stopping RB by inhibiting it. So let's read it. Loss of the normal cell cycle control is central to malignant transformation. Obviously, जो normal हमारा check points है उन्हें control हम कर रहे हैं, like 4 key regulators हैं, P16, cyclin D, cyclin dependent kinases, and RBC. RB. In cancers which are caused by certain oncogenic viruses, this is achieved through direct targeting of RB by the viral protein. And the classic example is the E7 proteins by human papillomavirus. So HPV produces two proteins, E6 and E7. Both of them have different mechanisms. E7, it binds to the active form of RB, so it cannot bind to E2F now. So it cannot inhibit the E2F transcription. The transcription is going to continue even though the DNA is mutated. RB is functionally deleted, which leads, because it has been sequestered by the E7 protein, it leads to the uncontrolled growth. Now coming to the next important short note which can occur, now coming to the next important short note for both again PG and UG examinations, which is T53. It is called as the guardian of the genome. P53 encoding tumor suppressor gene, which is TP53, is one of the most commonly mutated genes in human cancers. So there are three interlocking mechanisms, by which P53 controls a cell. P53 का main function है to control DNA damage. The DNA damage has occurred. G1S checkpoint पे RB ने आके रोक दिया है सेल को, but उस सेल को रिपेयर कैसे करना है is the function of TP53. That is why the name is the guardian of the genome. या तो वो quiescence, the cell will enter the temporary cell cycle, or there will be senescence, the permanent cell cycle arrest, or there will be apoptosis. This is programmed cell death, or normally कि वो सेल ठीक भी हो जाएगा, उसे नॉर्मल डिवीजन के रास्ते पे भी भेज दिया जा सकता है. A variety of stress triggers the P53 response pathways. Like सेल को जरा सा भी स्ट्रेस हुआ, एनॉक्सिया हुआ, कोई प्रो ग्रोथ स्टिमुलाई आ गया, कुछ डीएनए डैमेज आ गया, तो इस डीएनए डैमेज रिस्पॉन्स को कंट्रोल करती है P53. दैट्स व्हाई इंटेग्रिटी ऑफ द जीनोम को मेंटेन करके रखती है. द सेल विद डैमेज डीएनए जिसको रिपेयर नहीं किया जा सकता बाय P53, वो या तो सेनेसेंस में चला जाएगा यानी कि उसकी डिवीजन खत्म, और इट विल अंडरगो एपोप्टोसिस, दैट इज इट विल बी किल्ड. नॉर्मली एक नॉन स्ट्रेस्ड हेल्दी सेल में P53 का लंबा टाइम नहीं होता. इट इज शॉर्ट हाफ लाइफ. इट इज डिग्रेडेड बाय MDM2. MDM2 का फंक्शन ही यही है, उसको डिग्रेड करना P53 को. हाँ, ये सेल तो ठीक है, P53 की जरूरत नहीं है. इफ P50 is for a long time in a cell, that indicates उस सेल में कुछ DNA डैमेज है, because it is repairing it. That's why उसकी हाफ लाइफ प्रोलॉन्ग हुई है. MDM2 सबको डिग्रेड नहीं कर रहा, जब because so normally P53 की शॉर्ट हाफ लाइफ है and MDM2 is degrading it. Now DNA डैमेज के दौरान certain sensors activate हो रहे हैं, like ATM. अगर one of them protein kinases is ATM, that will catalyze modifications in P53. ऐसी modifications आएंगी P53 में कि अब वो MDM2 से रिश्ता जोड़ के नहीं रख पाएगा. It will release it from MDM2 and P53 की half life बढ़ जाएगी, and it will enhance its ability to drive the transcription of target genes, which are those target genes which are supposed to control the DNA damage. So these genes express neoplastic transformation by three mechanisms. The first is P53 mediated cell cycle arrest. It occurs late in the G1 phase. तो G1S पे आपका G1S checkpoint पे आपका सेल रुक गया है, अब G1 में लेट फेज में होता है. It is caused mainly by P53 dependent transcription of CDK inhibitor gene, which is P21. This is very important. P21 के थ्रू P53 ने सेल साइकिल को रोक दिया है. जो the P21 protein है, it will inhibit the complexes, जो cyclin CDK complex है, and prevent the phosphorylation of RB, thereby arresting the cells in the G1 phase. क्योंकि अगर RB phosphorylate होगी, which is technically hyperphosphorylate, उसकी inactive state चली जाएगी और सेल डिवाइड करने लगेगा. तो हमें अपनी RB को एक्टिव स्टेट में नहीं रखना, एक्टिव स्टेट में रखना है, यानी कि उसको हाइपोफॉस्फोरेटेड स्टेट में रखना है ताकि सेल की डिवीजन रुकी रहे. It gives the cell breathing time so that we can repair the DNA damage. The P53 protein also includes expression of DNA damage repair genes. If अगर ये DNA damage is repaired successfully, P53 will itself upregulate the transcription of MDM2, and this MDM2 will destroy P53 and relieve the cell cycle of the checkpoint of the block. The division will continue on. But if the damage cannot be repaired, the cell will enter P53 into its senescence, or can undergo p53 directed apoptosis. Second is p53 induced senescence. It is a form of permanent cell cycle arrest characterized by specific changes in morphology and gene expression that differentiated from quiescence or reversible cell cycle arrest. So this cell will not be dividing anymore. It has been decided. The mechanism of this are very unclear, but basically chromatin changes ऐसी आ जाती हैं which are no longer permissible for the cell to divide. P53 induced apoptosis of cells, थर्ड जो मैकेनिज्म है, with irreversible DNA damage. So P53 डिसाइड किया तो ना तो ये सेल नॉर्मल है, क्यूसेंस फेज में से नहीं भेज रहे, ना ही ये सेल ने बहुत ज्यादा डिवीजन करी है और फिर भी नॉर्मल है, बट इट्स नॉट वांटिंग टू एपोटोसिस भी नहीं कर रहे, तो सेनेसेंस में भी नहीं डाल रहे. थर्ड है कि इसका तो irreversible DNA डैमेज है इस सेल में, वी आर गोइंग टू हैव टू किल इट. It is the ultimate protective mechanism against neoplastic transformation. It is mediated by the upregulation of pro-apoptotic genes. अगर आपने फर्स्ट चैप्टर पढ़ा होगा तो you know pro-apoptotic genes and anti-apoptotic genes. तो pro-apoptotic genes को upregulate करती है p53 so that हमारा programmed cell death हो जाए और दो जो genes हैं BAX and PUMA. Now this is again a picture, a beautiful picture by the way from Robbins. If you look at the left side, basically normal cell है जिसमें p53 normal है और दूसरी है p53 खुद ही mutate हो गई तो कैंसर हो जाएगा. We are not discussing that yet. फर्स्ट, सेल को कुछ आयनाइजिंग रेडिएशन है, सेल में ही कुछ आयनाइजिंग रेडिएशन कार्सिनोजेंस म्यूटेजेंस ने कुछ स्टिमुलेट किया है डीएनए डैमेज. एक नॉर्मल सेल है जिसमें p53 नॉर्मल थी, डीएनए डैमेज हुआ या कुछ हाइपोक्सिया वगैरह नहीं, उससे p53 को एक्टिवेट किया, p53 विल गो एंड बाइंड टू द डीएनए एंड इट विल cause some transcription dependent and independent effects on its target genes. अगर सेनिसेंस में जाना है तो सेनिसेंस में भेज देगी सेल को, या फिर अगर उसको सेल को रिपेयर करना है तो हमें पढ़ा था P21 वेरी इंपॉर्टेंट सीडीके इनहिबिटर जो आपके कॉम्प्लेक्सेस को इनहिबिट करता है, जो डिवाइडिंग जो साइक्लिन डिपेंडेंट काइनेसेस और सीडीके साइक्लिन एंड साइक्लिन डिपेंडेंट काइनेसेस के जो कॉम्प्लेक्सेस हैं जो सेल को डिवाइड करने पे मजबूर करते हैं उनको ये इनहिबिट करता है P21. तो सेल का आपका अरेस्ट रुकता है, उस ब्रीदिंग टाइम में सेल को रिपेयर कर दिया जाता है. और दूसरी एक जीन है P20 के एडिशन 21 के एडिशन में GAD45, दिस आल्सो कॉजेज डीएनए रिपेयर. बट इन एनी केस अगर डीएनए रिपेयर नहीं हो पा रहा तो आपकी प्रो एपोटोटिक जीन्स आएंगी, BAX and PUMA, एंड एपोटोसिस विल हैपन. दिस इज व्हेन P53 इज इटसेल्फ नॉर्मल. P53 में खुद में कोई अबनॉर्मेलिटी नहीं है. बट इन केस P53 इज म्यूटेटेड या फिर देयर इज लॉस ऑफ P53, तब जो डीएनए डैमेज है P53 के जो डिपेंडेंट ये जो जीन्स थी जिनकी वजह से डीएनए डैमेज रिपेयर हो रहा था वो नहीं आई, दे आर नॉट एक्टिवेटेड. ना कोई सेल साइकिल में अरेस्ट आएगा, ना कोई डीएनए का रिपेयर हुआ, ना कोई सेनेसेंस फेज हुआ. सेल तो म्यूटेंट हो चुका है और उसकी अब अनलिमिटेड ग्रोथ होती रहेगी, एंड दैट इज व्हाट कॉजेज कैंसर. दैट इज व्हाई P53 is the most mutated gene in most of the cancers. Apart from tumor suppressor genes that we have already discussed, that is P53 and RB, there are other genes as well. And these are various mechanism of actions for various tumor suppressor genes, and we'll be enumerating them very briefly. RB, and then there's CDNK2A, VHL, of course, associated with RCC, STK11, associated with many cancers. Peutz-Jeghers syndrome, it is associated, SDHB, SDHD, and then Cadherin-1, gastric cancer and lobular breast cancer, TP53 obviously, BRCA1, BRCA2, MS2, MSH2, MLH1, MSH6 for HNPCC, WT1 for women's tumor, and MN1. That is all for tumor suppressor genes. Hope you liked the video. Let me know if there was something difficult to understand or any other topic that you would like me to cover. I will probably be making a video on the topics that are on the screen right now. And that is all for this video. Thank you.